Hello, my dear students. We continue our distant education, and we are sure that together we can overcome all difficulties that the current situation has put on us. I wish you all to stay safe and strong. Now I would like to start presenting our fifth lecture. I would like to tell you that because of the subject of this fifth lecture is too huge, I decided to subdivide it into two parts. So today you will listen the first part of this lecture. Today we will focus on mesenchyme, its differentiation, structural characteristics of cellular and non-cellular elements of connective tissue, classification, histogenesis of connective tissues, and connective tissue proper together with specialized connective tissue types. As we know, the connective tissue is mostly abundant type of tissue which we can find elsewhere in the body. Connective tissue, if we will look at the arm, for example, here we can find the bone which forms the skeleton, that is here, the bone tissue, then cartilage, which covers bone's articular surfaces of joints. Pay attention, that is cartilage. Then here we can see adipose tissue under the skin. Then we can find here tendons which join the muscle to the bone. Between bones we will find ligaments. Then loose connective tissue we can find under the epidermis, in the dermis component of the skin. Finally, in our blood vessels flows our other type of connective tissue, that is the blood. So it is mostly abundant type of tissue in the human body, and it forms continuum between the other types, epithelial, muscle, nerve tissue. By that way, it will connect all of these tissues into integrated body. By that way, connective tissue performs different functions. Here you can see that it is, first of all, that is the providing support for the organs. It forms the capsules and stroma of internal organs. By that way, it forms structural framework of the organ. Then it forms fascias, ligaments, aponeurosis. Then it forms the tendons. Then also, the bone tissue, cartilage form the skeleton, so that is a supportive function. Then it is the medium for exchange, that the nutrients, oxygen, waste products are exchanged between the blood circulation and the tissues of the organs via the connective tissue spaces. Also, connective tissue, it is a medium for inflammation, allergic reactions, so that is its functioning as defense, protection, because it reach in cells which have phagocytosing activity, immunocompetent cells. Also you can find the chemical components which will participate in immunological reactions. Then it is also storage of fat. Now we will talk about the classification of the connective tissues. Let's look here. That is previous classification which we have to take in mind. Here you can see the connective tissue proper, which include loose and dense connective tissue types. Then connective tissue with specialized properties that will include adipose, then reticular, hemopoietic tissue, another name is reticular tissue, mucous tissue. Then supportive connective tissues, these are the cartilage and bone. Also the blood also included here. But nowadays we have more modern classification which is accepted in most of the scientific literature. And here what we will see, let's look and we'll concentrate in this classification. Here you can see embryonic connective tissue, which include mesenchyme and mucous tissue. Then connective tissue proper, which include loose and dense connective tissue types. Also adipose and reticular tissue. The specialized connective tissues, which will include cartilage, blood, and bone. Now let's talk where they originate, from which source they take their origin. That is a mesenchymal tissue. Mesenchyme, as we told in embryology lecture, that mesenchyme, it is the embryonic connective tissue, and it will give rise to all of the connective tissue types. Let's look here. For example, chondroblast will develop into cartilage, adipocytes will form adipose tissue, fat tissue, fibroblast, that is prevalent type of tissue in connective tissue proper. These are cells which will form connective tissue proper. Then osteoblasts will develop into osteocytes, which will compose the bone tissue. Also hematopoietic stem cells, which also take their origin from the mesenchyme, and they will form all of the blood cells. Pay attention, all of the cells of the blood will develop from this cell, and this cell in turn develops from the mesenchymal cells. Also, pay attention to these two types of cells, which do not considered to be the connective tissue cells, but they also originate from the mesenchyme. These are mesothelial cells. These are cells, if you remember, this is simple squamous epithelium, which covers serous cavities, and the endothelial cells, which cover the blood vessels. So that is the origin of the connective tissue. And here you are looking at the histological section of micrograph of the mesenchyme. Here we can see pale staining, elongated, fusiform cells. They have processes in three-dimensional view, and they connect with each other to form network. That is the mesenchymal, but each of these cells, you know that they are highly potential cells that may develop into all of the tissue types. This type of the tissue is present just in embryo, and in adult human it is thought to be that present in pulp of the tooth. But in adult human, mesenchymal cells, these undifferentiated cells, will be replaced by other undifferentiated cells which we have in our body. These are pericytes, about which we will talk later. Mucous tissue. Mucous tissue, it is the main component of the umbilical cord. And also in subcutaneous epidermal tissue, we will have inside of the fetus body. It is jelly-like tissue, which include collagen third and first, a little amount of the first type. The mucocytes here, these are modified fibroblast cells. Because the extracellular matrix rich in hyaluronic acid, it attracts water. That's why it's jelly-like. Now let's go to the connective tissue proper. Here you are looking that it is one of its type, that is a loose connective tissue. It's also called areolar connective tissue. It fills the spaces of the body. It's present under the skin, under the mesothelium lining, inside of the internal organs. It's also present under the epithelium of the mucous membranes, which cover our tubular organs. It will form the lamina propria. And it is characterized by abundant ground substance and also tissue fluid. Inside of it, we will find cells and fibers, about which we will talk later. So the connective tissue, it includes cells and extracellular matrix. What is the extracellular matrix? It is the component which also present inside of the tissues, and its main components, these are ground substance and the fibers. The components of the ground substance, here you can see these are glycosaminoglycans, proteoglycans, and adhesive glycoproteins. Each of them, it has its own functions in the body. Let's talk about first of all the glycosaminoglycans. Here you are looking at this structure. These are polysaccharides, long-chain polysaccharides, and they include repeating disaccharides. From them, one of these disaccharides, it is uronic acid, and the other one is amino sugar. And because on the surface of these disaccharides, repeating disaccharides, we will have sulfate groups. Then they have also hydroxyl groups, which both of them have negative charge. So these molecules will attract sodium ions, and as you know, a high amount of the sodium in extracellular space will attract extracellular fluid. That's why these substances will attract the water and will be responsible for the gel state of the extracellular matrix. When these two molecules come to each other, the negative charges will repel each other, and that is responsible for slippery texture of the mucus. Then the glycosaminoglycans will attach to each other in the fashion of proteoglycans. So what are the proteoglycans? They resemble the bottle brush in which wire stem is composed of the core protein, to which attached glycosaminoglycans like bristles in three-dimensional arrangement. Here you can see these are glycosaminoglycans which attached to the protein core. By that way they will form proteoglycans. They are also different in shape, in size, and for example, if we are talking about the aggrecan proteoglycans, pay attention. Here one of the aggrecan molecule. These aggrecans mainly inside of the cartilage tissue and also in connective tissue proper. Via their core proteins, they attach to long chain of hyaluronic acid. By that way, there is formed aggrecan composites. So they're huge molecules which occupy the enough space in extracellular matrix. Pay attention, these are aggrecan composites. These molecules will be responsible for the gel state of extracellular matrix, and they will retard movement of microorganisms here. Then they will prevent also migration of the metastatic cells, will resist compression to the connective tissue. Also, they are responsible for slow disappearance of the aqueous bubble after the subdermal injection. So these are the proteoglycans. Now, we will look at here. Pay attention, this is aggrecan composite, just I told you, that is a hyaluronic acid, and these are our aggrecan proteoglycans, in which we can distinguish core protein, and these light blue colored chains, these are glycosaminoglycans. Pay attention that together with core protein they form proteoglycans. So that is a very important component of the cartilage tissue, remember. Then let's look how we will just review how these components are produced inside of the cells. Actually, connective tissue includes the cells which produce its extracellular matrix components. If we are talking about the connective tissue properties, are fibroblast cells. That is a nucleus. Here is encoded all of these components. Then rough endoplasmic reticulum, which will produce these proteins for export, as you know. Then the Golgi apparatus, and from there will arise vesicles which carry these proteins, their components, and they will release outside of the cell. Here we have to tell that the exception here, production of the hyaluronic acid, it is produced by integral protein which is present inside of the cell membrane, and it is hyaluronic synthetase, that is an enzyme which produces hyaluronic acid. And by the way, it forms inside of this protein, we have the channels through which hyaluronic acid is discharged outside of the cell. Here we also can see the other components. We have to take in mind that some of the proteoglycans are released outside, but some of them remain attached to the membrane of the cell, that is the decorin. That is syndecan. Pay attention, it has core protein and it has glycosaminoglycan chain. So this one also proteoglycan, if we have protein core, and we have also glycosaminoglycan, so that is our proteoglycan molecule. Here also you can see proteoglycan, the core protein of which, and the glycosaminoglycan chains which arise from this. Also we have to take in mind that the glycosaminoglycans cannot form covalent bonds with these core protein, and here there is link proteins are present, linker proteins, which will join these two components, two molecules to each other. Then some of these proteoglycans will participate in organization of the collagen fibers. Pay attention, that's collagen fiber, and in the organization also will participate our these proteoglycans and glycosaminoglycans. Here we will see members of them, as I mentioned, chondroitin 4 sulfate, 6 sulfate, dermatan sulfate, and each of them, they have their composition, localization, and functioning. Then here we also can see the collagen fibers and these aggrecan composites, proteoglycan composites. Here the hyaluronic acid chain, the green colored one here. And now we'll talk about the glycoproteins. Pay attention here. The types of proteoglycans, aggrecan, decorin, versican, syndecan, all of them, they also have their localization, functioning, their molecular composition. Their molecular composition almost the same, and the functions also almost the same. The proteoglycans which remain on the surface of the cell membrane, they serve for attachment of the cell to extracellular matrix components. Now we are coming to glycoproteins. Glycoproteins are adhesive molecules. These are like glue, which will stick all of the tissue components to each other. For example, let's look at this one. That is a cell membrane, as we know. Here we have proteins, integral proteins, pay attention, of the cell membrane, and they are extracellular matrix components, which we also now can recognize. What are they? This is a collagen fiber. And pay attention to glycoprotein. That is a fibronectin, for example. Fibronectin, it will serve for attachment of the integrin molecules on the cell membrane with the extracellular matrix components. The glycoproteins are of also several types. Here we will look at the types. For example, the mostly abundant from them, that is a fibronectin, which is present mainly in connective tissue proper. We have also laminin, then entactin, which was present in basal lamina, then chondronectin in cartilage tissue, osteonectin, osteopontin will present in bone tissue. All of them will be produced by connective tissue cells. For example, if we are talking about the connective tissue proper, that will be the fibroblast cells. Now let's talk about the fibers. The fibers which present in connective tissues, they are collagen fibers, elastic fibers, and the reticular fibers. The collagen fibers, they will resist, they will provide resistance of the tissue for the tensile forces. That is, these are strong, tough, and unbranched fibers. If we are looking under the electron microscope at the collagen fibers, they appear as banding, cross-banding light and dark lines, alternating dark and light bands, and they will repeat it through 6 to 7 nanometer intervals. In fresh state, they appear white. That's why they called white fibers also. It was estimated that they are tough and they are stronger, mechanically stronger than the stainless steel of the same diameter. The diameter approximately 10 nanometers, and pay attention, and they formed by fibrils. Then the collagen fibers, they produced by also connective tissue cells, and they are of 35 types in the human body. Each of these types differ from each other in the amino acid sequence inside of the polypeptide chain. Here we are looking that all of these cells, these fibers are produced by the fibroblast cells. These are connective tissue cells. How these fibers are produced? They, first of all, they produced in the rough endoplasmic reticulum because they need to be packaged and go outside of the cell. First of all, in the rough endoplasmic reticulum will be produced their polypeptide chains. They will called pre-pro collagen molecules. Then pre-pro-collagen molecules. These are polypeptide chains. Each of them will have on the end regions, they have propeptides, additional propeptides. Pay attention, they have pink color here. In the rough endoplasmic reticulum, on the next step will take place there glycosylation and hydroxylation. And here we have to mention that the hydroxylation of these molecules will need vitamin C as a coenzyme. That's why if there is some deficiency of vitamin C, it may lead to scurvy, that is the pathological condition, which will be firstly affect the parts of the body which with high turnover of collagen, that is mainly in periodontal ligament and in gingiva of the tooth, because that tissue becomes so soft because of the problems with collagen. So the normal collagen molecules will not produced, and the collagen molecules fibers will be not normally produced. That's why that tissue becomes soft and it will result in bleeding from gums. Even it may lead to loss of the tooth. So we are coming to next step. Then, as I told you, there will take place glycosylation. Then that molecules, pre-pro-collagen molecules, they are transported to Golgi apparatus. In Golgi apparatus, they will also undergo further modifications. They will attach oligosaccharide groups, and these molecules will be called procollagen molecules. Here, the triple helix will be formed from these chains. So polypeptide chains will will wrap around each other, and there will be formed procollagen molecules. Then, but why these propeptides are needed, you may ask. The propeptides are needed for which one? That is, procollagen molecule resembles as toughly bound ropes with the frayed ends. Here you can see these structures. Then these procollagen molecules are released via exocytosis into the extracellular space. But they released into the coves, the folds on the cell membrane. And these folds, these furrows will serve as molds to arrange the forming fibrils in the proper direction. We have to add here that these propeptides which present at the ends as a carbonyl and at both ends of the procollagen molecules, they form soluble molecules. That's why they prevent formation of the fibers inside of the cell. But when these molecules, procollagen molecules, are released outside of the cell, here will serve some enzyme, that is propeptidase enzyme, which will cleave these regions, these frayed ends. By that way will be formed molecules which will called, pay attention here, they are cleaved by the propeptidase enzymes, and the molecules which the length will be shorter, it will be 280 nanometers. These molecules will be called tropocollagen molecules. Then the tropocollagen molecules will self-assemble, pay attention here, and these tropocollagens, pay attention, that is procollagen with frayed ends, that is like toughly wound rope. These frayed ends will be cleaved. Can you see this one? And the tropocollagen molecules will be formed. Then the tropocollagens will align each other in staggered fashion. By that way will be formed fibers, fibrils, then the fibers. The staggered manner will be created by the covalent bonds between the lysine and hydroxylysine molecules inside of the polypeptide chains. We also have to tell with you that the mainly amino acids which are present inside of the collagen molecules, this prevalent here glycine, also present lysine, proline, or hydroxyproline and hydroxylysine. These are prevalent amino acids in the polypeptide chains of collagen molecules. When we will look, pay attention here, these tropocollagen molecules between these head and tail regions will remain gaps. These gaps, they align each other in neighboring rows. By that way, we will see these gaps through these alternating regions will be approximately 67 nanometer in length. When we will prepare the slide for electron microscopy, we use osmium salts that is a post-fixation procedure for the preparation of the slides. And these osmium salts, these heavy metal salts, will deposit it in these gap regions. That's why we will have appearance of alternating dark and light bands when we will look at these collagen fibers. Pay attention. Here we now will look. Pay attention, amino acids, their diameter 1 nanometer, then tropocollagen 30 nanometers length. Fibrils, we will have 1 micrometer, and the fibers, they will be more than 10 micrometers. Between these fibril forming collagens, they are mainly present first, second, third type, fifth type, and 11th type, that they are prevalent in the body. We have also non-fibril forming, network forming collagens. These are 4th type and the 7th type. On those molecules, the procollagen, the propeptides which are present at the ends of the procollagen molecules, they are not cleaved. That's why they will not form the fibers. Then we are coming to elastic fibers. Elastic fibers, as implies their name, they provide elasticity to the connective tissue. Their main components, the elastin protein and fibrillin proteins. When the elastic fibers are produced, they are produced by the fibroblasts of the connective tissue and smooth muscle cells in the wall of the blood vessels. Here, first of all, produced the microfibrils. Microfibrils are formed by fibrillin 1 protein. Pay attention, these microfibrils. Then, during formation of the elastic fiber, then the elastin protein will be deposited into the between these microfibrils, so the elastic fibers are formed. So at the periphery of the fiber, we will see microfibrils, and in the center, we will see elastin protein. Elastin protein also produced in proelastin form. Inside of this protein, we will find mainly lysine and isolysine, hydroxylysine amino acids, also glycine. Elastic fibers we can find in the connective tissue as slender, unbranched, long molecules. But in some cases, they may form more coarse bundles. For examento in the ligamentoflavum of the vertebral column, they will form ligamentoflavum, which is elastic and strong, by the way. Elastic fibers also form elastic fenestrated membranes in the wall of the elastic arteries. There is very important, the elasticity of the wall of those vessels. You know that they carry blood from the heart. That's why there is needed their elasticity. How they become so elastic and they may be stretched? Pay attention to this structure. Between 4 chains of amino acids in elastin protein, there are formed between the lysine amino acids of these 4 chains. There are formed some cross bonds, cross links, and 4 polypeptide chains will be bound to each other via these links. And these bonds will be called desmosine and isodesmosine links. By the presence of them, the elastic fiber may be stretched approximately 150% of their resting length without breaking, and then they return to the previous length. So here you can see the relaxed state, and here you can see the stretched state of the elastins molecule. Then we are coming to the basement membrane. So, and something about the reticular fibers. Reticular fibers, they are also have the structure like in collagen fibers, but they are thinner. They form fibrils, and that is the third type of collagen which forms reticular fibers. They mainly present in reticular tissue, in which we can find in hematopoietic organs. The basement membrane, as we know, that is the base for epithelial tissue, that is where the epithelial cells rest, and that is separate the epithelial tissue from the underlying connective tissue. It is an interface between the epithelium and the connective tissue, and this one is called basement membrane. Pay attention. Basement membrane include two components. That is a basal lamina. Here, that one we can study under the electron microscope. The basal lamina and lamina reticularis here. Lamina reticularis and basal lamina. If basal lamina is produced by epithelial cells, so the reticular lamina is produced by connective tissue cells. If we will look at the basal lamina under the electron microscope, we will see that it composed of the electron lucida, lamina lucida. It is electron lucent layer, and it is composed of glycoproteins, entactin and laminin, which form here network. They, in one side, they attach to the epithelial cells, to the integral proteins of the epithelial cells. Here you remember they are present mainly integrin molecules, and on other side they attach to other components. Now we will look at them. Now lamina densa, next component. It also have 50 nanometer thickness, and it is mainly composed of the collagen 4 type. It form here network. As you remember, collagen 4 type, it doesn't form fibers. So here they will form network, and this network will serve as molecular filter for passage of the substances from between the epithelial and connective tissue components. Then we will talk the lamina densa. The lamina densa, it includes the fourth type of collagen. It forms here network, and on both surfaces, this network of collagen 4th type covered by perlacan, that is proteoglycan, and we know that this perlacan, it is proteoglycan which carry heparan sulfate on its surface, that is glycosaminoglycan, which have negative charge. That's why it will serve as filter for negatively charged ions, so the basement membrane will not permit entrance of negatively charged ions through it. That is very important in, for example, filtration in kidneys. Then also we have to know that lamina densa is anchored to the reticular lamina via anchoring fibers. These are collagen 7th type. Let's look. Collagen 7th type, it is called anchoring fibers. Here also present fibrillin and also present fibronectin. It also serves for binding of the lamina densa to the lamina reticularis. The lamina reticularis is formed by, produced by connective tissue cells, and it is formed by collagen 3rd type, which form reticular fibers. Also here present 7th type of collagen, which form anchoring fibers. Here we can also find the 13th type of collagen together with the microfibrils. What are the functioning of the basement membrane? First of all, as I told you, it is a barrier, it's filtration barrier between the epithelium and the connective tissue. Then pay attention here, you can see it. It also will simulate differentiation and proliferation of epithelial cells which rest on this basement membrane, will serve as a guide for migration of the cells during wound healing. Also, basement membrane will facilitate the formation of the cell polarity. as we know, epithelial cells have polarity, and that is the basement membrane plays here important role in the establishment of the polarity of the epithelial cells. Here also we can see that the functions also become thickened due to increased collagen and laminin production. And for example, in diabetes mellitus, the basement membrane of small blood vessels, especially those in the retina and kidney, becomes thick and thicker than in normal state. So that are the functions of the basement membrane. Now we are coming to the connective tissue cells. When we now told about the extracellular matrix, let's now talk about the cellular components of the connective tissue. Here you are looking at the schematic representation of the connective tissue. Here you can find all of its components, the cellular and non-cellular components. We have to know that the cells of the connective tissue are subdivided into two categories. First of all, they are fixed cells and transient cells. Fixed cells, they are developed and stay in place in connective tissue, and they perform their main functions here. They include fibroblasts, that is fibroblasts, pay attention. Then we have here adipose cells of two types, unilocular and multilocular. These are these cells. Then we have pericytes. Pericytes are located on the surface of blood vessels. Pay attention, that is a blood capillary, and on its surface, like a spider, surround this blood vessel from the periphery. That is a pericyte cell, and differentiated cell, by the way. Then here we are looking at the macrophages. These are also fixed cells. And finally, here you can see the mast cell. That is the other next type of the fixed connective tissue cells. And we have also transient cells. These are actually the blood cells which circulate within the bloodstream, and upon receiving the signal from the connective tissue, they will leave bloodstream, enter the connective tissue, and perform their functions there. As you can see here, one of the cells migrating outside of the blood vessel. These are actually white blood cells. These are neutrophils, eosinophils, basophils, lymphocytes. All of them, these belong to the blood cells. Then we are coming to each of these cells. For example, the fibroblasts. These are mostly abundant and widely distributed cells of the connective tissue. They are tissue-forming cells. They produce all of the components of the extracellular matrix, fibers, ground substance components, all of them, they are produced by these cells. If we will stain the slide with hematoxylin eosin, we cannot distinguish these fibroblast cells because they have very pale staining cytoplasm. What can we distinguish? They're just the nucleus, which is clearly visible in hematoxylin staining slides. The fibroblasts mainly line along the collagen fibers. They surround these collagen fibers. The fibroblast cells present in two forms. That is, it is active cell, which is called fibroblast, and quiescent state. It is called fibrocyte. But these are transitory. That's why it is difficult to distinguish them. Fibroblast cells, when they actually non-dividing cells, but they may start to undergo division, may enter cell cycle when they receive signal, for example, during wound healing. On that case, the fibroblast may be also differentiate into myofibroblasts. Myofibroblasts, these are like fibroblast cells, but they include also actin and myosin complexes. These are contractile filaments. That's why they will have ability for contraction, and it is important in wound contraction. Myofibroblasts will resemble smooth muscle cells, but unlike them, they will not have covered by external lamina. So these are myofibroblast cells. The pericyte cells, these are, as I told you, surround the blood vessels, blood capillaries and venules. They are undifferentiated cells, so when they receive signal, they may differentiate into smooth muscle cells, endothelial cells of the blood vessels, and other cell types. They are like mesenchymal cells. The pericytes, also called perivascular cells or adventitial cells, they also include actin myosin complexes. They have ability for contraction, and in that case, they will regulate the lumen of the blood capillaries which they surround. Then next type, adipose cells. Pay attention, we have two types of them. Adipose cells, they differentiate from the undifferentiated cells, and they are non-dividing cells. We have unilocular, as you can see here. Here the cytoplasm of the cell is occupied by one large lipid droplet. The nucleus is replaced to the periphery of the cell. These cells will form white adipose tissue. That one are multilocular adipose cells. They will have polygonal shape, and the cytoplasm will be filled with mitochondria that carry cytochrome pigment. That's why these cells will form brown adipose tissue. Then we are coming to the macrophages. Macrophages, actually, we have two populations of macrophages. First of all, fixed macrophages. They are characteristic for the organ where they reside. For example, Kupffer cells in liver, microglia in central nervous system, or then the dust cells in the respiratory system. So they are resident cells of those organs, fixed cells. But we have also migratory macrophages. These differentiate from the monocytes that migrate along the blood vessels, and when they receive signal from the tissue, they migrate to the tissue and differentiate into macrophages. So what we have to know, macrophages, they have actively phagocytosing cells. They phagocytose particular matter. This may be foreign invaders, maybe waste products of the tissue degeneration. Then they process those antigens and present the fragments of those antigens on their surface. That's why they also called antigen presenting cells. So these are the macrophages which have irregular shape. They may have philopodia, some extensions which are needed for the movement, for the performing of the specific functions of phagocytosis. Then we are coming to the mast cells. The mast cells, or tissue basophils, the cytoplasm filled with the granules. These granules contain mediators, very important mediators of inflammation and allergic reactions. Okay, then about these cells we will talk later. Here we can see also the transient cells of the connective tissue. They are blood cells, these neutrophils, then eosinophils, even migrating neutrophil we can see here. Then the lymphocytes, pay attention. The monocytes we also can find here. Finally, plasmocytes, plasmatic cells or plasmocytes. They are cells which are differentiate from B lymphocytes when they undergo activation by antigenic challenge. They activate to become plasma cells. Plasma cells, these are important components of immunological reactions, mainly in humoral immunity. They will produce antibodies, immunoglobulins. Pay attention to the cytoplasm. Near to the nucleus, we have light area. That is area where located Golgi apparatus and the centrioles of the cell. So that is morphological distinguishing feature of the plasma cells from the lymphocytes, how we can distinguish them. Then, okay, now let's talk about the, let's look at the electromicrograph representations of these cells. First of all, fibroblasts, which I just told you about them. Then the macrophages, here we can see, which have irregular shape. They have lysosomes in high amount. They have processes like pseudopodia or lamellapodia or philopodia. Then mast cells with granules inside of them, nucleus in the center. That is the multilocular adipose cell, which are different, several droplets which carry lipids here. And in the center, we can find the nucleus. In the cytoplasm, there are mitochondria, so they have high amount of the mitochondria. Here we are looking at the cell types. Here what we have to pay our attention. Just I told you that macrophages, they are of two groups. They may belong to fixed cells and maybe transient cells. But all of these macrophages of the body, they are produced from they develop from the monocytes of the blood. All of them belong to mononuclear phagocyte system. These are cells which belong to this system. All of them have phagocytosing activity. So, for example, the macrophage here, sinusoidal macrophage, Kupffer cell in liver, alveolar macrophage in lungs, then fetal placental antigen presenting cells, Hofbauer cells in placenta, then pleural or peritoneal macrophages in serous cavities, osteoclasts in bone, microglia in central nervous system, Langerhans cells in epidermis, also oral mucosa, in genital epithelium, in esophagus we also can find them. Fibroblast-derived macrophage we can find in the lamina propria of mucous membranes, dendritic cells in lymphoid nodes and spleen, multinucleated giant cells, they are found in pathological granulomas. That means that when the antigenic challenge, the macrophages may fuse with each other to form multinucleated giants that you have to know. Now let's look at the mast cell and how it undergo activation. Actually, mast cells, they are active participants of immunological reactions in immediate hypersensitivity reactions. They carry on the cell membrane the receptor for Fc fragments of antibodies, immunoglobulins E. When the exposure to some foreign antigens, for example, bee venom or pollen or some drugs, in the body produced antibodies against those allergens, these immunoglobulins E, then they come and bind to the surface membrane receptors of the mast cells. And during second exposure to the same antigen, to the same allergen, these allergen will bind to these immunoglobulins E, which are now on the surface of the mast cells, and they lead to their activation. When it activates, we will see that inside of this cell we have granules that contain primary mediators of inflammation. What are they? These are chondroitin sulfate, histamine, heparin, eosinophil chemotactic factor, neutrophil chemotactic factor, aryl sulfatases. These are primary mediators of inflammation. So activation of the cell will lead to increase of the calcium concentration inside of the cytoplasm, and it will result in discharge of the granules. That process will call degranulation of the mast cells, and all of these components will be released into the extracellular space and will cause the effects there. Then it also will activate phospholipase C, that is the enzyme, and it will cleave, it will lead to formation of the arachidonic acid derivatives. They will be leukotrienes, thromboxans, and prostaglandins. They will call the secondary mediators of inflammation, and they do not conserve, they do not concentrated, they do not deposited inside of the cell, but they are released when it is needed, when the cell is activated. So the release of these components into the extracellular space will lead to what? Finally, the permeability of the blood capillaries will increase. The capillaries will become wider, and it is resulted in the leakage of the plasma components into the extracellular space. It will lead to edema, increase of extracellular fluid amount. Then it will also lead to release of the attracting neutrophils, then other leukocytes into the site of inflammation inside of the tissue. Finally, we will have all of the signs of inflammation that you will study it from the pathological anatomy course. It will lead to edema, to rubor, the reddishness of the tissue, then increase of the temperature in the local region if it is local response. But if it will be generalized, all of these components may lead to anaphylactic shock. For example, this component, leukotrienes, thromboxans, and others, also histamine in turn, they will also lead to constriction of the smooth muscles in the wall of the respiratory system. It will lead to bronchospasm. It will lead to increase of the mucus production in the wall of the respiratory system. So increased amount of mucus, and this mucus becomes so slippery, concentrated, and bronchospasm finally may lead to stop of the breathing, and it may lead to death even. Now we are coming to here something about the adipose cells. As we know, we have two types of adipose cells. These are unilocular and multilocular. That is, unilocular adipose cells, they participate in the storage and production of the triglycerides. They participate in the metabolism of triglycerides. When in the blood there is high amount of chylomicrons, these are a result of absorption of lipids in the wall of the intestines, then very low density lipoproteins are produced in the liver. These products inside of the capillary of the near to the adipose tissue, they circulate. Then near to the adipose tissue, they will leave bloodstream, and here they will be broken down by lipoprotein lipase to free fatty acids and glycerol. These components will be transported into this cell, fat cell, adipose cell, and there will be produced triglycerides, which will be stored in droplets. And if, for example, during fasting, if there is deficiency of fatty acids and lipids in the blood circulation, it will activate the adipose cells, and they will cleave these triglycerides to glycerol and fatty acids. Here will be acting hormone-sensitive lipase. This lipase will break these components, and glycerol and fatty acids will enter blood circulation. So the adipose tissue will perform functions in lipid metabolism. Also, adipose cells will produce some lymphokine. They will produce some components which called adipokines. They are, for example, leptin and other substances which will also control lipid metabolism. Now let's go to the next type of the connective tissue proper. We now finished the loose connective tissue, and now let's look at the dense connective tissue. Dense connective tissue characterized by the prevalence of the fiber components. Pay attention here. The main component here, these are the fibers. If they arranged in parallel in some order, they arranged in parallel to each other. On that case, we will have dense regular connective tissue. For example, we can find it in tendons, in ligaments, aponeurosis, fascias. Between the collagen fibers, they will form bundles actually. Between the bundles, we will see individual fibrocytes, fibroblasts, elongated cells that align themselves between the collagen fibers. Also here we can see the loose connective tissue interstices, number 3 here, that are supporting components which will carry blood vessels and nerves to supply these dense connective tissue elements that will form individual organs, for example, tendons. If we are talking about dense irregular connective tissue, that one here the collagen fibers will form bundles that have random orientation, and so they will form like meshwork. Between them we can find the connective tissue cells, here mainly present fibroblast cells, macrophages, mast cells in small amount. Also the blood vessels we can find here, so that is dense irregular connective tissue, which is mainly present in the dermis of the skin, also in capsules of some organs, in mucosa of the internal organs. Here we are looking at the adipose tissue. Pay attention, the mesenchymal cell, they also develop from the mesenchymal cells. They will differentiate in two directions, multilocular and unilocular. That is unilocular white adipose tissue. Multilocular cell will form brown adipose tissue, which is present mainly in fetus. And finally, reticular tissue. Reticular tissue, as a reticulum, it means network. It's formed by reticular cells, the fibroblast-like cells, and the reticular fibers. These are formed by the collagen of third type. So that is the end of the lecture. Dear students, thank you for your attention.